Good afternoon, everyone, and welcome to the Plant Habitats and Wildlife Interaction session. Dr. Debra Ayres and I will be co-chairs for this session, and we will be introducing our guest speakers.
Is this number 26? Is this number 26?
Is what number 26?
Okay, go ahead and do your intro. Sorry.
This first talk is the effects of a fire break on plants and wildlife at Pine Hill, a biodiversity hotspot. You can put the first slide on. Okay, good, thank you.
Yeah, fortunately, the first front row said 21.
Okay, I'll keep talking then. So this presentation is divided into three parts, two speakers. The first speaker is Mario Klip, who will introduce the study. Dr. Klip is a wildlife biologist, a bear biologist, now working for the California Department of Fish and Wildlife, not on bears. He manages over 100,000 acres of wildlife areas and ecological reserves, a lot of fire, and he enjoys collaborating on multi-dimensional projects like this one. He will be followed by Jenna Meyer. Dr. Meyer is a retired professor from Sacramento City College, where she taught botany and field ecology and has seen many of her students here today. You might have learned botany from her. Now she is an active volunteer with the El Dorado Chapter. In addition to leading field trips and weed pulls, she has been conducting research on Pine Hill, which she will discuss today, and is involved in the genetic research of Pine Hill flannelbush with, it wants to reverberate, with Professor Dan Potter. Mario will then return to discuss the effect of the fire breaks on the animals. So, Mario.
Thank you, Debra. Thank you for the introduction. And just to clarify, I'm still working on bears too. In fact, earlier this week I was at a bear conference talking about how to keep bears out of trash and all kinds of other things. So what we're going to do today is actually an interesting study, because many studies are focusing on just one element, one piece of an ecosystem. And this study is really focusing on how we can do adaptive management really in the field, especially as an agency, which is not always easy to do. So before we just assumed that putting a big fire break into an area, we actually talked to each other and said, okay, well, what kind of impacts are we really expecting for wildlife? What kind of impacts are we expecting for these the large number of endangered and special status plants in the area? Before I further get into it, I just want to acknowledge that Molly Caldwell is not here today, but she was also part of this research. So I'll talk about the study area a little bit, just to kind of give you an idea as to where it's going. I think many of you in the room may already know where Pine Hill is, because I suspect this is not the first talk that you've seen on Pine Hill, but I'll go into that a little bit. The study questions and some of the logistical details as well that we ran into trying to do this research. And then we'll go into the details as laid out on the slide. So first of all, the study area. You can see here, and I don't think there's a pointer here, but on the left-hand side of the slide, you see the study area. And the study area is entirely landlocked. So in other words, there is private property, small parcels all the way around it. In fact, even for us as an agency, we are not able to get in without going through someone's backyard. The area had not burned for a very long time, and it was decadent with manzanita and all kinds of other plants that were really hard for some of the wildlife to be able to get through, but also for the endangered species of plants that were trying to make a living there as well. So some of the photos that I'll show you in a minute really show you how dense of a wall of chaparral was actually there. And one of the things that we ran into when we wanted to actually bring CNPS out, and without this collaboration, this project wouldn't even be possible in order to avoid any damage potentially to these plants. When we took them out and we tried to say, okay, well, listen, let's look whether we can find any plants, there was no way to even get in. So the first thing we did is that we had people build 4 trails around the entire perimeter that were spaced evenly apart so that we could actually see what was growing there. That was kind of the first kind of little hiccup that we ran into. There were several others, but that was probably the most time-consuming one. So why is it relevant to talk about this in wildlife? Many areas that are kind of anthropogenically impacted generally have a very incomplete assemblage of wildlife. And what was interesting, and I'll show you some videos that maybe some of you already saw earlier, we were surprised to see that essentially the assemblage that you would expect in the foothills is actually completely present there. The other thing that I think is relevant to bring up is that this is a very unique area. It has unique soils, gabbro soils, and as a result, it's home to many plants that don't find a home anywhere else in this world. So this is what it kind of looked like, and we used Cal Fire. It was a fantastic collaboration during the times when they were available. So obviously, when you're trying to do burning, we don't do that in August because even though there might be forest fires in August and that might be more natural, obviously it's way too dangerous. So in that sense, when we were doing our treatments, we had to do it when these crews were available to us. So this is what it looked like. So you can see obviously the piles in front of that very dense wall of chaparral in the background, and essentially firefighters coming from a CDCR, so California Department of Corrections camp, that were helping with this work. So what are the questions that we tried to address? We tried to understand the survivorship of Cyanotus odriki with and without fire. We also tried to understand how vegetation responded to clearing and clearing plus fire, since many of these plants are obligatory and need fire to be able to germinate. We try to understand exotic versus native species diversity and their cover. And will the communities return back to its native chaparral? And then we also looked into how does this clearing actually affect wildlife in the area. So just a couple of images to kind of give you an idea as to what these intrepid volunteers from the CNPS were dealing with. It was a massive amount of work. And for those of you who are interested, there was a paper published 2 years ago on this topic, and there's a lot more details in it. I'll put it in the end as well. But an enormous amount of work went into this. And of course, and this is something that I rarely see. I mean, I have small children, and if I give them a cookie, they look extremely excited. But I have not seen people that excited by finding some of these plants. I mean, it's like calling people flying in to see these plants. I mean, unbelievable. So it was truly magnificent to see some of these photos, and every time I was able to, to go out with them in the field, it was just such an exciting thing. And even though I studied bears, my dad is a botanist, so he always tried to instill me an interest in this, and it's been a phenomenal project to work on together with CNPS. So with that, I just wanted to ask Gina to come up to talk about the botanical component of this study.
And can I have the laser pointer?
Oh, yes.
Thank you. Oh, thank you so much, Mario, for that great introduction, and thanks for including our chapter of CNPS in this study. It's, even though it's been an incredible amount of hard work, it's been very rewarding, I think, for all of us that are involved. So I want to thank you for that. So for the plant studies, for our vegetation studies, Mario mentioned and how the crews put in that firebreak, and that firebreak was constructed in 2017. The vegetation studies that we're talking about were on the west slope of Pine Hill, and so the firebreak there had also been cleared in 2017. But we didn't start our studies until the spring of 2019. So our volunteers, Tal and Bill, there are standing within the firebreak, and beyond there you can see that impenetrable chaparral. So on the west portion of Pine Hill where we were working, the chaparral has a very dense shrub layer, as Mario mentioned, mostly, dominated by whiteleaf manzanita. And it has a ground cover that is dominated by creeping sage, so Salvia sonamensis. But there is also on the ground cover, we have the invasive exotic annual false brome, which you can see here in the right, in a clearing of the intact chaparral, that it's there, and it's also in the firebreak as well now. Okay, so you saw that the material that was cleared from the firebreak was actually burned in piles. So after those burnings, then we had small areas that had been burned. So essentially, we had replicate burns. This doesn't happen very often for you. We're able to avoid pseudo-replication, if you've ever heard about that. So we had these small burn piles, and so we had these small plots. These became our research plots. And then we paired those with the same size plots in the cleared area that was adjacent. So behind this little fire plot, or burn plot as we call it, then there is a cleared plot right adjacent to it where we took our data. And these are small circular plots. So a couple of things then. We were focusing on one of the endangered species on the west side of Pine Hill. So this is Pine Hill ceanothus. And we wanted to see how it responded to both the fire and the burning and the clearing of the firebreak. So what we did is we marked, we marked with nails, and we counted and we measured the sizes of all of the seedlings in the plots of Pine Hill ceanothus. So that was one part of the study. The other was we wanted to know how the plant communities responded to the two treatments, the clearing of the firebreak and the burn pile, so how they responded to the fire. So what we did is we identified all the species and all the plots, and we estimated their plant cover. So data collection, as I mentioned, started in 2019. The 2019 results were actually published in the paper that Mario was referring to, and we'll give you that information in the end. But we have continued the study the last couple of years in 2021 and 2022. You can probably guess why we weren't out there in 2020. We ended up with 8 pairs of burned and cleared plots together. And so for the cleared plots, they were cleared in 2017. So therefore, we have data collected from the 2nd, 4th, and 5th year after the clearing. We have 5 of our burn plots that were burned in 2017, and hence we have data from those same 2nd, 4th, and 5th years. But 3 of our burn plots were actually burned in 2018. And so for those, we have data from year 1, year 3, and year 5 after the treatment, after the burning. And I just wanted to mention how we marked our Pine Hill ceanothus seedlings. Here's our little arrow. See that little red dot? That's a painted nail. So every time you see one of these little painted nails, there's a seedling there. In amongst that lovely yellow star tulip, Calochortus monophyllus, if you know that one. Okay, so we'll start off with what we found out about Pine Hill ceanothus. Turns out that Pine Hill ceanothus is one of those obligate cedars we've been hearing about in the chaparral sessions. Remember that the rare plants were marked and were avoided. And so Pine Hill ceanothus in the cleared areas of the firebreak are extant. They weren't affected. And so they will be in some of our cleared plots. But in the burn plots, we just have seedlings, because if the Pine Hill ceanothus had been in the fire plots, they would have been burned, but they were avoided so that wouldn't happen. Okay, so some results. Here are some results from the 5 years, since we have 3 plots that have 1 year, 3 years, and 5 years data, and then our 5th year data, and then we have plots that have 2nd, 4th, and 5th year data. That means we have 5 years of data for our Pine Hill ceanothus seedling study. So the Pine Hill ceanothus seedlings in the burn plots are in red here, and so the number of seedlings per square meter. And so we started out with a really good flush of seedling emergence in the first year, but that number of seedlings decreased over the next 2 years. And then they started to increase again, where seedling emergence then began to exceed mortality. I will mention that we did take a look at the Pine Hill ceanothus seedlings in the fall of 2019, and a number of seedlings had died during that summer drought. So we know that we did get mortality because of summer drought. Over the years, though, the seedlings that did survive grew. So this is height, so average height over the years. And so our average heights then, of course, increased. And so those surviving seedlings did grow. So here is a few of the little seedlings. Here's one that's rather larger. And these little white spots are not flowers. They are actually the nails, the red paint faded away in the sunlight. So, yeah, I said, where are those flowers? No. Okay. Okay, so the fact that we had continuing seedling emergence in the last 2 years after the fire, we can see in this stem and leaf plot. So actually in this plot here, this was plot number 6. This year we counted 89 seedlings in that plot. It's just incredible. And half of them have a diameter of equal to or less than 9 centimeters. So we had a lot of very, very small, lots of really small seedlings. And that suggests that they are relatively newly emerged seedlings because they're small. We did have one, though, that was almost half a meter in diameter. So, so this, I think that this really shows the adaptive ability of Pine Hill ceanothus. And as long as the fire intervals are not too short, as we learned about from John Keeley yesterday, that we should have continued persistence of Pine Hill ceanothus, and also if the if droughty conditions don't continue too long. Okay, so the next thing I'll talk about is the plant community responses. And so what we have here in this first slide is just the percent cover of the plants in the plots over time, over those 5 years. But the left graph is in the burn plots, and the right is in the cleared plots and the cleared only plots. And we see that, as you would expect, that over time the plants in the burn plots would increase in cover, and they did. This also occurred from the 2nd to the 4th year in the cleared plots as well. Of course, the cleared plots were not completely bare because the clearing occurred no more than 30 centimeters below, above the ground. So a lot of plant material was left. And so we did have quite a bit of plant material left in the cleared areas, but we got more in year 4. There was growth that occurred. But from the 4th year to the 5th year, there was a decrease. I'm suspecting that that may be due to our continuing drought, and maybe we had some die-off. Whoops, wrong way. Okay, so we can, let me go back here for just a second. So the blue are the native plant covers, and the red on top are the exotic species cover. So it's interesting that both in the burn plots and in the cleared plots, that exotic species do contribute a fair amount of cover in both treatments. We can't tell a huge amount of information about the differences in communities from just the total covers or even the covers of native versus exotic species. So we need to look a little bit further to see if there are if in fact there are differences between the burn treatments and the cleared treatments. So one way of doing that is the community coefficient. And community coefficients work by determining the number of shared species and the cover of those shared species in the two treatments. And so if you have a coefficient less than 50, that indicates that you have two different communities. If we just look at, in blue here, over the 5 years, the blue bars indicate community coefficients that is just dependent on the number of species that are shared, the number of species that are shared. And they do share a fair proportion of all the species that were found in the plots are shared between the two, as one might expect when they're right next to each other. However, if you look at the cover of those shared species, that differs between the burned and the cleared treatments. These are the orange bars over the years. Those are definitely less than 50. So that indicates that we do have differences in the communities in the two treatments. Another way of looking at this is with a multivariate technique called cluster analysis that Debra Ayres ran. And we see from this that the early fire treatment years, so that's years 1, 2, and 3 in the fire plots, they cluster together. Maybe that's not surprising that they would share species and would cluster together like this. But what is interesting is that the late fire treatments cluster apart from the early years fire treatment. So this is years 4 and 5. That means that the covers of the various species changed over time and changed over time so much that they differed from what we saw in the early fire treatments. Then also what's interesting is that all the cleared plots that cluster together, now that's not surprising, 6, 7, and 8 refer to the 2nd, 4th, and 5th year following clearing. And so these are clustered together, but they cluster closer to the early fire treatments than the late fire treatments. So that's interesting. So we wanted to ask ourselves why. Why would this be the case? What species may be involved in determining this or causing this pattern? So what we did is we took a look at the individual species that had high cover in one or both of the treatments. So we'll start with a couple of dominant species in our chaparral, whiteleaf manzanita and creeping sage. And so our Arctosophilus viscida is one of those manzanitas that does not sprout. It's an obligate cedar. In the cleared plots, you can see which is in green. You know, the green is the cleared plots, that we have increased cover over time in the cleared plots. This is due to seedlings. They were chopped in the cleared areas. Those mature manzanitas were cut during the clearing. So what has come in are seedlings. And that's an interesting thing, that they're coming in the clearing. The red bars, of course, are the burn plots. We do see a few of the manzanita seedlings in the blue burn plots, but not as many as we do in the cleared plots. This tells us that the seeds are not fire dependent. So for Salvia sonamensis, creeping sage, it was already present in the cleared plots because it creeps. It's low on the ground. It's very, very prostrate. So it was not cut during the clearing. It was already present, and so it was of fairly high cover in those plots, in the cleared plots. It decreased from year 4 to 5. Perhaps, again, this is due to continuing drought, and there was some die-off of our creeping sage. We also have, you see, that there is creeping sage in our fire plots a little bit more over time. This is not due to seedlings. We saw virtually no seedlings of the Salvia sonamensis. This is because it creeps in from the side. It lives by its name. So it creeped into those plots vegetatively. So we can see this in the next slide. So here in this plot, plot number 4, these are the little manzanita seedlings growing quite nicely and being very happy. And over here in plot 5, we can see the creeping sage. That's this. It's kind of hard to see, but it's there. And creeping into this plot. And we saw quite a bit of it in this plot in this year, in this spring. So what about other types of plants and how they do their reproductive thing? Well, seeds responding to fire. This is, of course, our Ceanothus rodericii. Our pine heel ceanothus does that. So this is kind of a reiteration of what you've already seen here, that pine heel ceanothus seedlings emerged and grew in the burn plots and were already present. The mature shrubs were already present in the cleared plots. And for this one, peak rush rose, Crocanthamum, Scoparium. It used to be Helianthemum, if you know that name better. So we saw just a little bit of it in the cleared plots, but it responded to fire quite nicely, and it grows so quickly. It just really takes off. So it was producing quite a bit of cover, more than 25% by the 3rd year. In the cleared plots, we didn't see much. What was present was already there and continued to just be small, probably because of competition, and didn't spread much. So here is Crocanthamum in one of our burn plots, in plot number 8. Boy, it really took off here. It really took off and contributed more than 50% cover in this plot. Next, let's take a look at those lovely resprouters. In our chaparral, we have Yerba Santa, and it didn't come in by seedling much, although that is definitely possible, but boy, did it ever resprout like crazy in the burn plots in red here. We saw a little bit of it in the cleared plots. And then for Lepikinia calycina, or pitcher sage, it also resprouted in response to fire. It didn't cover too much. This is only 3.5% by the 5th year. But pitcher sage is kind of patchy in our chaparral, as is Yerba Santa. You see some here and then maybe over there, but it's patchy. It's not very continuous. So we'll show you some pictures of those guys. So here's Yerba Santa in our plot number 7. It just really just grew so quickly. And then pitcher sage here. There's just one little plant here. Didn't produce much cover, but it resprouted in this burn plot number 6. But also it can come up by seed. Here's some seedlings that I took a picture of in another burned area at Pine Hill. So it also comes in by seed. So it's one of those facultative guys. What is concerning, however, is the high amount of cover of the invasive exotic annual false brome that we're getting in both our burn plots and in our cleared-only plots. It is a California Invasive Plant Council rating of moderately invasive. And it's not that the clearing and the burning caused the false brome to come in. It was already present. Remember that slide that we had at the beginning? It's already present in the intact chaparral. So it's not the clearing's fault. It's already there. We actually think probably where it came from to come into our cleared perimeter is perhaps actually from adjacent residential areas that may have come in that way. So it's a problem because it dries out really quickly, and then it forms thatch that could provide fuel for continued fire. So an interesting thing is if you take the 7 species that I just talked about, all those 7 species, and you run the cluster analysis on just those 7 species, then you end up with a pattern that's almost completely identical to the pattern that you get if you run the analysis with all of the species. So very good correlation between the two. That tells us that these 7 species and the cover they're producing in the burned versus the cleared treatments are causing this pattern. That's what's causing the pattern. So it's interesting that those species are so important in determining these communities. So just quickly, I want to mention we are continuing our firebreak study in the south side of Pine Hill, focusing on Phremonodendron decumbens, which is Pine Hill flannelbush. It is both a resprouter and an obligate fire seeder, if you will. It requires fire for seed to germinate. So we'll be looking at that study later on. One last thing. Oh, geez, running out of time. I'm just going to skip over this, and we'll go right to Mario's wildlife talk. Here, I'm going to leave this here.
All righty then. So a few things that Gina just said is that we were cutting about this high, and the primary reason that we did that is because of the species that we cared about, T&E species that we cared about. Only one of them was taller than this, which is the Phremonodendron. So we did not want to accidentally cut it. And since we were using inmates, and even though we tried to train them, it was thought to be safer to do it that way. So around the perimeter of Pine Hill Preserve, we put 12 cameras. 9 cameras were essentially in areas that were treated, and 3 cameras were used as a control. And what we tried to do is create an unduplicated set of photos. So what we did is that any animals that were showing up on cameras, or the same species, would show up again. We would actually remove that, and also any animals that would show up on adjacent cameras within 1 hour of the first photo, we would remove that as well. And I don't have time to go into all the model testing, but obviously we did a VIC analysis and looked at season and camera location, time of year, all those types of things that you would normally do as part of a model selection. But I think that what I'll do is I'll focus on the cliff notes that are kind of most interesting. So we had almost 3,000 photos of critters that we found. And what's interesting is that, I think maybe you remember from the beginning, is that this area is actually closed to the public. And what we found is that this firebreak was actually inviting people that were living as neighbors to us to actually start using it almost as a hiking trail. And that's something that we're still addressing right now, because it also, unfortunately, was an additional downside. Someone also thought it was a great idea, now that it was a little bit more open, to grow marijuana. And then we had some unhoused individuals that also thought, hey, this is now great to access the area also. So that's a little bit of a downside when you make things open. And we had someone going all the way up the chain to complain that we had removed their privacy. So they care more about privacy than potentially their house burning down, which is sometimes a little bit hard to fathom if you live in California, but sometimes not everybody has the same opinion of things. So let me get into the results. So I already mentioned earlier, and I don't have time to go into all the details of how we did the exact model selection, But what I think the most important thing is to understand is, was wildlife benefiting from us doing a firebreak, yes or no? And the way we were trying to do this is to understand whether the detection ratios that we were actually seeing, were they higher in treated areas, so treated, burned, as well as cleared, or in areas that were essentially intact chaparral areas. And what you can see here is that for all species here identified, you would actually see that they're more easily identifiable in areas that are cleared. Now, for deer, that might be something that you might expect, since they're large-bodied animals, and as a result, they might have a hard time getting around. But especially for smaller animals, we did not quite expect that. What we also looked into is some of the carnivores that we were seeing. And again, we see that same trend occurring here as well. Now, this may all sound somewhat intuitive, but what we were actually seeing, and this is, I think, what's interesting about this research, is that we looked at the plants and we looked at the cover, and then what we saw is that in the areas where we had a higher percent cover of palatable species of plants, that we would see a higher density of herbivores in that area. And then as a result, we also saw a cascading effect where now that there's more herbivores, that we actually saw more predators showing up in these areas as well. So there's a positive correlation between squirrel detection rates and some of these mesocarnivores. So you can see that here depicted in these graphs, how strong that connection is. And that's really interesting. And one of the things that I'll do at the end is I'll show a short video, and you actually see some of these mesocarnivores running around with small prey in their mouth. One of the other things, and that's just a little side note, when you do something like this in an anthropogenic landscape, so you're basically creating a firebreak, and you are essentially seeing that it becomes more attractive to wildlife, there's also some side effects. I just talked about the cannabis and some of those things, but there's another side effect that happens as well. We started noticing bear, and we started noticing mountain lions as well. And some of the folks that are immediately bordering us, they think it's cool to have a couple goats or a couple alpacas or something like that. And unfortunately, the mountain lions found those as well. So we had some questions about that. It's not something that we can tie together, but it's certainly something that you have to consider when you do this kind of work, where you make a habitat more hospitable for species, that there might be additional components that you may not think about. So let's see, how much time do we still have? Are you at about the whole 5 minutes? All right. So what I'll do is I'll show a short little video. It's only a minute of the species that we saw and some of the interactions that we were able to witness, playing, mountain lions, all that kind of stuff. So hopefully you enjoy this too. It's not playing, Mr. Soundman. That's what I thought was going to happen. All right. Is this showing? All right, Mr. Soundman.
Just play your desktop. Just share your desktop.
Okay, go ahead. It's not playing for me in the presentation.
No, I'm just going to share the desktop.
Yep. Thank you. We clapped too early.
Doesn't mic? Yes. It's playing. I have a few PowerPoints. Okay. Here, you want me to move on?
Oh, I'd love to show it. That'd be a couple minutes because we do.
Okay. Well, then I need to move it out right here. Did I just lose the computer?
result, computer has to be restarted?
No.
Maybe while we're waiting for the video, we can take questions? Yeah? Or, I think there's a few more minutes, right? Sounds good. Yeah, I mean, he's restarting the computer, so that's going to take a minute, I think. Yeah. Are there any questions? Is there someone with a microphone that's...
Yeah, that would be me.
Oh, that would be you. All right.
Just repeat back
But
the
I'll
question.
repeat the question back so that it can be captured.
Do you have any observations on post-fire recovery after wildfires in that area?
That's a good question. Sorry, is there any evidence of post-fire recovery in that area? Right?
I'm curious, particularly in terms of the seedling density, whether it differs the pile burns versus the natural fire, largely because the pile burns have a very unnatural fuel structure, too, doesn't they?
It's true.
Well, a lot hotter than a wildfire would. So I'm curious, do you have comparisons with wildfires?
We don't.
So the question is, the fires, the way they were used in the study, are not mimicking natural fire regimes, and they're burning much hotter. And as a result, is there any difference between natural or more natural fire regimes? Potentially. So
I don't know of any species, I mean, for this species, I don't know of anybody that looked at a natural fire. It's really rather confined.
I think that what I'll add on is that initially when we talked about this, we were expecting essentially no survival, right? Because we were feeding sticks, or they were feeding sticks on these areas where they were burning, because we actually ran out of space in some areas because it was so thick, not to hurt any other plants. And I don't want to paraphrase Debra's words because I'm not a botanist, but if I'm saying it correctly, and you can nod, I believe that we saw a delayed response in some areas. But no response. So in other words, as a non-botanist, the way I would paraphrase it is that even though it was an unnatural event, it still allowed plants that are obligatory seeders to actually be able to reproduce.
But yes, we should look for an area like that, but I don't want a fire to burn next to my house, which is where I am.
There is a fire in Salmon Falls. I don't know whether there's CA notice there, though.
No, there isn't.
The computer is logged out right now.
Oh, are you still going? Still going.
You wanted to take that question?
Thanks for that question, John. I actually do have the data from work I did 25 years ago, and it never even occurred to me to pull that data up and compare it to the data we just got. So thank you very much for the question.
Stuck?
Can't tell.
Are there any other questions, or are we done?
Were you all surprised actually were you surprised that some of the plants that you might have expected to come up from seed after the fires did not come up? I mean, some of the plants that you might have thought because they're chaparral plants would respond to the fire did not. Were there any surprises there?
I would like to address that. The dominant chaparral species, the dominant manzanita, doesn't like fire. Were we surprised at the responses of some of these plants to fire? They live in the chaparral. For many years, I've assumed that Arctostaphylos viscida was primed to germinate by fire, and we saw zero germination after fire. Didn't like it, and much slower germination in the years following fire than we did in the cleared plots. And it makes me wonder if the dispersal and the digging by, as Tom Parker said, by little rodents, either did or did not help it. So I was very surprised. And another dominant, Salvia sonamensis, is killed by fire, no seed recruitment. How does that work? it grows in from the side. So it was very surprising for me to see this. So,
All right, it looks like the video is now working.
Thank you, Mario and Gina. Our next presentation is native plant and pollinator interactions at the Pine Hill Preserve. Thank you.
Go ahead, keep going. Okay, are you done introducing him?
Thank you.
Yeah. Thank you. Don't face the speaker.
Okay.
And talk loud.
And our next speaker is Landon Eldredge. Landon is, he graduated from Northern Arizona University with a bachelor's degree in biology and a certification in wildlife management. He has worked with native plants and pollinator interactions on Pine Hill Preserve managed lands since 2014 and actively works on vegetation management projects to enhance habitat for rare plants. In addition to his work on Pine Hill Preserve, Landon also volunteers with the BLM's Folsom Lake Veterans Crew to help fight wildfires around the country. Welcome, Landon.
All righty. Let me see. Okay, cool. Am I loud enough? No? Okay. If I'm not loud enough, somebody throw a tomato or something. Okay, so yeah, I'm Landon Eldredge. I work with BLM as a biological technician, like Graciela said, and I'm going to get into the native plant and pollinator interactions at the Pine Hill Preserve. Okay, cool, thank you. So I'll breeze through the intro part. Pine Hill is about 5,000 acres of managed lands. It's a cooperative conservation effort between 10 different agencies. We have about 10% of California's native flora and 8 rare plants. That's the purpose that the preserve was established for, and the gabbro soils of the rescue series are what give rise to the unique flora in our region. Vegetation management is kind of our bread and butter. So as Mario and Gina were just talking about, we'll have crews come in and cut the dense chaparral. This creates openings for native forbs and the rare plants to move into. And then ideally we'll burn it, and that has great benefits for some of the plants that require fire for germination. But it also removes the vegetation from the ground, which creates more bare soil, which benefits some bee species, which I'll talk about in a little bit. Sometimes we'll chip or masticate vegetation as well if burning's too hard to do because of the forever narrowing burn window in California. Other stuff we do guided tours. That's what the first picture is about. And then we'll do native seed collections. We have more than 240 so far, and rare plant and post-burn monitoring as well. So now the native bees and flora of Pine Hill Preserve. So first off, I'm kind of gonna separate. So most native bee pollination is done by solitary reproductive females, and this is different than how honeybees or bumblebees do their stuff. And we wanted to focus on native bees in the study because we know there's a great diversity in California, and they also have unique relationships that non-native bees or other pollinators might not have with the native plants. So yeah, going back into what makes native so great, the coevolution of it. So we knew that the native bee diversity was great and that most of them are generalists, and bees are a largely visual kind of group of insects. At least when it comes to seeking their pollen and nectar resources. So some of the more obvious stuff, like the picture on the right, with all the woolly sunflower down there at the bottom. We knew that bees would be seeking out, or we figured bees would be seeking out something like that, but the less descript and kind of more cryptic species like the fritillaria on the left, those unique relationships, we wanted to find out more about those as well. And we also know that there's many bees that are thought to be pollen specialists, so we really wanted to key in on those relationships also. How does non-native vegetation fit in? It's not good because it often creates monocultures if it'll move into an area and it kind of outcompetes the natives, and loss of floral diversity is the primary cause of native bee decline. Okay. So for the collections, the bulk of what we've done was in between 2014 and 2020, and most of them were done through direct observations. So this was seeing the bee on the flower itself and collecting it then, taking down the information, what, where, when, et cetera. And then we also did some supplementary sweep netting and blue vein and malaise trapping as well. And the supplementary specimens are housed in a museum collection at the BLM field office. So getting into the results, and some of these numbers are kind of rounded with a plus sign because we've done a few collections since the bulk of this data was put together, but we haven't had them identified by specialists yet. So you'll have to bear with me for that. And if you can't see this graph real well, it has the plants across the top and going down in columns. It's just different bee species that were found visiting these flowers. So some highlights. We had more than 145 total bee species, and 143 of these were native, 2 of them were non-native, and we collected from at least 65 plant species, and at least 60 of these are native, 5 were non-native. And then now narrowing our focus a little bit more to the rare plants. This table's laid out the same. The rare plants were visited by 68 of 145 of the bee species that we collected in our studies. The Wyethia reticulata brought in the most bee species out of anything we collected from, with 40 different species. And the Fremonodendron, which was the only species that we really had much pollinator On the previous research, there were 2 bees that composed 98% of the visitations to the flower, and one of those we didn't record, but we do have a few more species that weren't mentioned in that previous study, and so that the hope is that they're kind of supplementing where that other one left off. And then lastly, the Gallium californicum sierrae is left off of this graph because we didn't detect any bees on it while we were doing the collections. And then now narrowing to just one rare plant. This is the Calistesia stebbinsii, and it's a federally and state listed as endangered species. In the situation I'm going to talk about, it's growing in a wildfire burn scar. Population is probably one of our strongest and seems to be holding steady. And it's regularly visited by this Diadasia species that's in the middle of the flower. And then the relationship between these two is really close. These were the types of relationships that we were hoping to find out more about in our studies. So the Diadasia nests, or it's a morning glory pollen specialist. It nests in the bare soil within the Calistesia population, and the hairy males will hang out inside the flowers in the afternoon, and then you'll regularly see the females flying from one flower to another. And we only have a few records of this Diadasia species outside of this general area. So you can see that the two are tightly linked, and then we use the research whenever we get additional research from folks like Mario and CNPS and others, and try and see where it fits back in with the management practices that we have. So again, our vegetation management, we're cutting holes in the vegetation, which allows for the forbs to grow like Calistesia, and then burning these fuels allows access to the bare soil for Diadasia and other ground nesting bees to nest. All right, and then how do honeybees fit in? Not real well. We didn't see a lot of them out there on the preserve, but they were there, and this is probably because being non-native, they haven't co-evolved here, and they're excluded from certain resources. So, for instance, with manzanita, because of the shape of the anthers and how they release their pollen, honeybees don't have access to this, but you will occasionally see them nectaring on the flowers. So this will have a competitive cost with native bees that seek that nectar reward. If not, they're going for the pollen as well. But the honeybees also might be supplementing some plants with additional pollination. So with coffeeberry, our Frangula californica, most of what I've seen on them is the European honeybee. And so we take this information into consideration when apiculturists will contact us and they're asking us if they can place honeybees on preserved lands. And then, okay, cool. Poor pollinators. So as far as the preserve was concerned, not all species under the total pollinator umbrella seem to be particularly important to us, but they do have their purposes, of course, and their ecological functions, but their protections as well, like that of the monarch or the masked bees, which is the bee below it. Their protections protect their habitat, and those benefits will spill over into places like the preserve. And then lastly, looking forward and back on the work that we've done so far. So some of the more remote areas of the preserve that were harder to reach due to the impenetrable chaparral that they were talking about earlier. some of these areas, like the one that most of these pictures are from, this one has a riparian band running through it, as well as some serpentine soils, which are less common on most of the preserve. And so this will host a whole slew of different flora in that area, and then nesting resources as well. And there's research out of Flagstaff that supports the idea that nesting resources, not just floral resources alone, is actually the limiting factor in what bees might be present in a certain area. And and lastly, we just got contacted recently. A PhD student from UC Davis is interested in the microbiomes of native bees, and specifically that Diadasia species that specializes on our calisthesia. So looking forward to hopefully doing future research with them and then working it back into our management practices. Thank you. Any questions?
Do you know about the Diadasia? Is it like any specific conditions for ground nesting?
I don't know that other than bare soil, and I only know of it at that one site, but hopefully when this researcher comes in, we'll get more information on that. Thank you.
So you were looking at just visits or the visitors, you weren't really looking at pollinators, right? Or whether they're actually pollinating.
Correct.
Repeat the question.
Okay. Yeah, sorry. So the last question was, are we able to measure whether or not they were actually pollinating, or are we just looking at visits? And we were just looking at visits. Go again.
For that calisthesia, is there another pretty common frequent visitor besides that Diadasia?
No, there wasn't.
Not at all.
There were other visitors, but not with any regularity. And at other sites, other than this one where I saw that nesting population of the Diadasia, there's not very regular visitors either. Go ahead.
The native bees' affinity for bare soil.
Mm-hmm.
I mean, thinking back before Europeans, where would there have been bare soil?
Sorry, were you not?
No, I mean, I know on my own property, maintaining bare soil takes work. Just curious.
So where would the bare soil have been back before humans were kind of manicuring to keep it around? More or less. Or clearing, yeah.
I mean, how so productive it would be producing flora.
So my guess is that this would be from wildfires that are in the area, and as the plant diversity in certain areas kind of ebbs and flows as wildfires come in and then vegetation grows back over, that's how these bees were getting access to certain areas. Also, the potential for game trails, creating these small openings where there's bare, compact soil, which is also something that certain bees like. Maybe they'd move into areas like that as well, or tree fall, yeah. Mm-hmm. Go ahead.
I was wondering, did you observe flower visits by other insect groups like flies, like wasps or beetles, at a high number?
So did we see other insects visiting the flowers other than just bees at a decent amount, I guess? Sorry, I'm not paraphrasing well. But no, we didn't see them very often. There were certain plants where there was more of that going on than others. In the in the calisthesia, actually, outside of that site where the Diadasia is, there's a lot of beetles that would show up in the flowers, but we just kind of tried to narrow our focus to bees for the study, so we didn't look into those too much. Go ahead.
With respect to
I haven't thought about that. I think they, from what I've seen online, the species itself is relatively widespread. It's not unique just to like Pine Hill Preserve or anything like that, but it does specialize on morning glories in general. But in our area, it's specializing. We don't really have other morning glories. Thankfully, we don't have the non-native ones that grow off the side of the road. So it happens to be specializing, my guess, just on our morning glory.
Okay, we have an opportunity to catch up on time.
Cool.
Thank Landon again.
Our next speaker is Dr. Steve Peterson. He's an entomologist who owns and operates Foothill Bee Ranch in Forest Hill, California. No, he did not burn up in the recent fire. Where he raises blue orchard bees and consults with almond growers on pollination with these bees and is involved in solitary bee research on almonds and blueberries.
Thank you for that introduction, Debra. Let's see. Okay. Right. So I'd like to talk to you a little bit about the blue orchard bee, which is a native Osmia species here in the western U.S. And I was interested in looking at whether it could be found in the Sierra foothills, and if it could be increased so that excess bees could be used for pollination in things like almonds and cherries in California. So a little bit about the biology on this species. Well, it's commonly called the blue orchard bee or orchard mason bee. Mason because it uses mud in its nests. These are not official common names, so but it is commonly referred to both of those names. It nests in pre-existing holes in wood. So in nature, it can be found in dead trees where there's beetle borings, but it will not chew out its own hole. It has to find a pre-existing hole. So this is often a limiting factor in their population dynamics. They have just one generation per year. They're solitary. They're not social, so each female is reproductive. Females don't have any contact with the next generation. In other words, once they lay their egg on the pollen provision, there's no more contact between the mother and her offspring. So very different than, say, the honeybee, which is a social insect. They don't make wax or honey. Probably about 40 years ago, they were identified by the Logan Bee Lab, a USDA lab, as a potential valuable crop pollinator because they visit a wide variety of plants and because they nest above ground in holes. They can be moved to where they're needed. And lastly, they are native to the western United States. There is an eastern subspecies, which might actually be a separate species. They're currently looking at the taxonomy of that. And there may be a subspecies in the west as well, a southwestern subspecies. Okay, in commercial pollination, they're currently being used to pollinate things like almonds, cherries, pears, berries. They're highly efficient at pollinating, and this is because each female is gathering pollen on every flower visit, unlike the honeybee, which may be going after nectar on many of its visits. They carry the pollen dry on their body. They have a brush of hairs on the abdomen called a scopa. The pollen is packed there, but it's held dry, whereas with the honeybee, they moisten the pollen and pack it on their legs, and it's a little less available to fall off onto the next flower. So they're highly efficient. It's been estimated that about 300 nesting females is enough to pollinate an acre of almonds. And that would replace one honeybee hive. So the recommendations are to go with one honeybee hive and 300 nesting females. Because it's a male-dominated sex ratio, about 1,000 bees will yield about 300 females. Currently, the bees that are pollinating almonds, the blue orchard bees that are pollinating almonds, are mostly coming from Washington, Oregon, and Utah. But it's known that they're in California. I was interested to see if we could find them easily in the Sierra foothills and if they could be propagated to large numbers using similar methods that they use in the Pacific Northwest. So there's different nesting materials that they will accept. They'll use solid wood blocks, and that's the upper left there. You can line that with a paper straw to be able to remove the nest and manage the bees. And it's important, if you're managing these bees, to be able to open the nests and remove any predated cells or parasitized or fungal infected. Otherwise, these things can really take over a nest and bring down the reproduction rate quite a bit. But they'll also accept grooved wooden boards, paper tubes on the lower left, cut reeds, and even plastic nests will be accepted. As far as preference goes, solid wood is probably their favorite, but the most difficult to manage. Second favorite would probably be reeds and paper tubes. But the grooved wooden laminate nests are becoming probably the most commonly used in commercial pollination because the bees like them and they can be opened easily and the cocoons managed that way. So for nesting, they prefer a hole that is 6 inches deep and 5/16 in diameter, or 7 to 8 millimeters. When they're nesting, they'll go and collect pollen and nectar. One pollen load will usually take about 75 flower visits. And then and she'll bring that back to the nest, remove the pollen, and add a little bit of nectar to it to moisten it, and then go on additional trips. To complete one provision to be able to lay one egg, that would take about 25 such trips. And so one provision in order to produce one offspring would represent about 1,875 flower visits. A female can live up to 6 weeks, possibly longer if the weather is cool. When it is rainy and cool, they'll tend to just hole up in their hole and wait for warmer weather. When it's like that, sometimes their lifespan gets a little bit longer. They can lay up to about 32 eggs. They can complete one provision for one egg per day, sometimes 2 or 3 in really ideal conditions. And then mud is used between the cells and to plug the end of the hole. And seen in that picture are some bees gathering some mud or moist soil. So here's a look at inside of a nest, and this happens to be a reed that's been split open. The opening is to the right, and to the left, which is out of the picture, is a node. They need the hole to be plugged at the end. They don't like to be able to see light through the tunnel. But when we split that open, you see back at the back are 2 cocoons where bees hatched and came out. One of those females returned and made a nest in that reed. And you can see what they do first is make a mud backing, and then they gather pollen and gather enough to be able to lay an egg on it, and that's the egg that you see there. The eggs in these bees are much, much larger than honeybee eggs, or a honeybee can lay like 1,000 eggs per day. This bee just is laying 1, 2, or maybe 3, so she invests a lot more energy into each egg, and the egg is much larger. And you can see different sizes there. Towards the end, the last 3 are quite a bit smaller. Those would be male provisions. So females are larger. They get a larger pollen provision. This nest is a little unusual. You usually don't see that many females. Oftentimes it's 2 or 3 females and then maybe 5 or 6 males. So that's a particularly good nest. Oops. Okay. Like I said, the bees have 1 generation per year. It takes the larvae all summer, and they become adults in the fall, and then they go through wintering. So they spend the winter as a fully developed adult bee inside a cocoon. They are spring flying bees, so that adaptation, wintering as an adult, allows them to emerge rapidly and go right to work on early spring flowers. They can be held in a refrigerator. The cocoons can even be washed. They're sealed up tight. They have a waxy coating. But sometimes you have spider mites or fungal spores, and it can be beneficial to wash them in a weak bleach solution and then dry them, and you can remove a lot of those potential threats. So my question was, could we find the bees readily in the Sierra foothills? So I approached BLM to do a small study. The sites that I looked at were Case Mountain in Tulare County and Kanaka Valley in El Dorado County, which is a part of the Pine Hill Reserve, correct? Yes, right back to Pine Hill. Okay. Okay. And it's near Folsom Lake. So here's just a couple of pictures from Case Mountain. It's a beautiful, very large BLM property. It's got a lot of oak woodland. It's got grasslands. It's got chaparral. So it's got tremendous plant diversity, wide range of elevations. So it was pretty likely to be able to find blue orchard bees there. And Kanaka Valley is similar, a little bit lower elevation, and it's also got grassland, oak woodland, and some chaparral. These blue orchard bees, or Osmia lignaria, I'm not a plant person, but this is a plant meeting, and so just a little taste of of some of the plants that they'll visit. And this is a bee is quite generalist. So western redbud, manzanitas of different species, Pacific madrone, deerbrush or ceanothus of various species, even species that are wind pollinated like oak, maple, and willow, they will gather that pollen. It is a nutritious pollen for them. California blackberry, coffeeberry, gooseberry, and then a wide range of forbs, annual wildflowers like Phacelia is one of their favorites, Nemophila lupinus, Calencia, another favorite, Calencia, and Penstemon. So when I'm looking at habitats, I kind of look for some of these, and if they're present, there's a good chance you might find Osmia lignaria. So the way I did the study was to construct nesting units out of half-gallon milk cartons that I spray painted black and cut off the top, and then use paper tubes in there. And the dark color, the previous studies have shown that they don't prefer bright or reflective kind of nest boxes, like white is not preferred. Most other colors are acceptable, and black is good because it kind of hides in the environment, particularly in a place like Case Mountain where there's lots of hikers that go by, and so if you can hide them a little bit, all the better. When you come, I typically put those out in February and then come and pick them back up in May. And this is what you kind of hope to see when you come to pick them back up. And I did 2 kinds of these milk carton nests, one with a plywood separator, because the recommendations are to have a little bit of distance between the holes, but you only get 25 per carton that way. And so I also tested just bundling the tubes up all together, and obviously that's acceptable too. And so you can get a lot more holes per unit that way. So I did this for 3 years at Case Mountain, starting in 2017. And so for 2017, I just put out 1,645 holes in bundles of either 25 or 75, depending on the nest arrangement. I did not release any bees with them, so it was just bare holes. And like I said, holes can be a limiting factor for their population. So when I came back in May, there were 1,481 completed nests, which was surprisingly good. I didn't really expect that many. Once I opened those all up and cleaned them, I found about 11,878 bees. So I calculated a reproduction rate when I put bees out to see how well they multiplied, so that wasn't applicable that year. But I also took a sample of the nests and categorized all the different mortality factors, which I'll get into in a moment. Had 6.4% mortality on those cocoons. So the next year, I didn't put all those bees out at Case Mountain. There was a study that USDA at the Logan Bee Lab needed some bees from California, so I donated some bees to them. So I was only putting out 7,700 bees that year with more holes this time, 4,137. This time I got 3,200 nests completed. This was all basically in the same region of the Case Mountain Preserve, and it probably took up an area of about 60 acres, so a relatively small portion of it. So that small area can support a large number of bees. And so once those nests were opened up, it turned out to be 24,100 bees, a reproduction rate of over threefold, which is outstanding, really. Mortality went up a little bit, though, had some other insects get in there, and I'm going to show you what those factors were in a moment. For the final year, again, I didn't put back all those bees. Some of them went to a pollination study for the USDA, but I put out 6,144 with over 5,000 holes. This time came out to about the same number of nests and a similar number of bees produced. But also about a threefold increase in bees. So really excellent reproduction there. And a mortality of 17.7%, so about the same there. At Kanaka Valley, only ran this in 2018 and 2019 and at a smaller scale. Started off with 500 holes with no bees in 2018. I got 98 completed nests and almost 500 bees. So the next year, 100 of those bees went to another study, but I was able to put out 400 of those bees, or 395, and I got back 313 nests. So I was able to increase them from there, which is over fourfold rate of reproduction. At the end of both of these studies, all the bees returned to BLM and were released in 2020. So just a quick look at the mortality factors that I was able to categorize. A pollen ball is just where the egg did not develop. Sometimes you can't find an egg. An egg may have not been laid there, or the egg shriveled up and died for some reason. So we call that a pollen ball. Sometimes you find a dead larva where most of the pollen was consumed. Tricrania is a blister beetle. This beetle lays its eggs on flowers, and then the first instar larva grabs onto a bee leg when a bee comes visiting, and it gets taken back to the nest, and then that larva develops by eating the pollen and the bee larva that was there. So it's a kleptoparasite. Sapidja is a wasp species that waits around solitary bee nests and then goes into the nest when the female is gone, and she lays her eggs there, and then that Sapidja wasp develops on the pollen. Chalkroot is a fungal disease. It's Ascosphaera. It's common in solitary bees. And finally, Stelis, or Stelis, however you want to pronounce it, is a cuckoo bee. It's, again, like Sapidja, it waits for the solitary bee to be gone, and then it goes in and lays its egg on the pollen, so kind of cheating. You can see Case Mountain, The first year, all of those things were fairly low. They did increase a bit, particularly tricrania came up quite a bit. And I think when you start having larger and larger number of bees in a given area, some of these predators can find them a little easier. But this is a good reason why it's important to open your nests and remove those things. They're easy to spot once you open the nest. Their cocoons look quite different than the bee cocoons. Kanaka Valley was a little different. We did not have tricrania at all. One year, that first year, quite a bit of chalkbird though. And I don't really know why it was so high that year, but I was able to bring it back down the next year. So. And here's just a look at these things. Tricrania, Stansbury, blister beetle, quite a pretty beetle, but they can be devastating on solitary bees. Sapidja is that little wasp there. Stelis montana is the cuckoo bee. It actually looks a lot like a osmia bee, but it's smaller. The head is smaller. And then chalkbrood, the fungal disease. It infects the young larva, and once it gets to a late instar, the larva dies and its body is then filled with spores. The bee behind it has to chew through that, and it gets covered in spores, and then it's going to pass it on to its nest. From that, you might get the impression that it's very easy to raise blue orchard bees, and it can be. But since that time, I've been also raising bees on private property, and these are some of the reproductive rates that I've seen. 2018-19 were pretty good years for reproduction in general, but you can see the last 3 years were not. 2020 was particularly bad. It was very dry in the spring. I think moisture timing is very important there. And so just to conclude, Osmia lignari bees can easily be found and propagated in the foothills. Year-to-year propagation results are highly variable. The part that I did at BLM looked very, very good, but since that time on private properties, it hasn't been quite as good. And mortality factors include 3 kleptoparasitic species and a fungal disease that are important to control. So that's all I have. I don't know if there's time for questions.
Yes, thank you very much. Maybe if people have questions, they can catch you at the end of the next session. Thank you very much, Steve. Our next presenter is Christopher Schwind, and he's an ecologist working for Creekside Science, and he's primarily focused on the conservation of Bay Area butterflies and their associated habitats. These habitats include many local endemic plant species as well, and he's also actively engaged in the bolstering of the native plant populations. His presentation is the Keeping Mission Blue Butterflies in the Loop inus, and I have to be very careful at how to say this because my first language is Spanish, so welcome, Chris.
All right, so today I want to talk to you guys about one of the very cool projects that I get to work on with my Creekside Science team. They're kind of hiding over here in the corner. And yeah, so first let me introduce the Mission Blue Butterfly. This is a pair of them here. Sure. Is that okay? Is that better? Okay. So here's a pair of them, obviously having a good time. These are a federally endangered butterfly. They're not state endangered. Many of you might know that the State Endangered Species Act doesn't cover insects. So, little factoid there. The largest population of them is on San Bruno Mountain, where we're working currently, and they utilize 3 lupin species as their host plants, Lupinus formosus, elberfrons, and variecolor. Primarily the elberfrons, but the other 2, as we'll come to see, are also really important. So this is San Bruno Mountain. Beautiful spot. If you guys haven't been there, go there. It's really an incredible park. It's both a state park and a county park. And in the background there, is that laser? Okay, I brought a laser pointer. Oh, well, we'll toss this one out. This one I think might be got it at Army surplus. I think it might be like anti-tank or something. But ooh, look at that, right? Okay. Anyway, so yeah, this is the city behind us. And from up on top, you can get views of both the San Francisco Bay and the Pacific Ocean at the same time. So this is a cool spot. This has been a stable area of conservation since the 80s. It was the first established habitat conservation plan. For those of you who aren't familiar with that, it's essentially a community getting together and making a plan that sort of balances needs for development and conservation at the same time. At one point, this was slated to all look like the surrounding sprawl. So thank goodness for the Habitat Conservation Plan that protected this area in perpetuity. So yeah, as you can see, it's also surrounded by this urban environment, and so it's really like this island of all these cool species. There are a lot of serpentine soils here, so there's a lot of plant diversity, there's a lot of butterfly diversity. This is a really, really critical little island left in the urban landscape. Also, largely, this was protected because of the endangered mission blue butterfly, as well as some other butterflies. The mission blue was listed in 1976, and that was one of the primary reasons why people had some pull to really get this land protected. So anyway, the serpentine soils here are now under threat from non-native grass encroachment, as are most serpentine soils around the state. And a big reason for that is nitrogen deposition. So all the cars and everything are basically dumping nitrates onto soils everywhere, and especially in a place that's in the middle of a big city like this. And they're fertilizing the soil. And so these serpentine soils that are typically low in nutrients, are suddenly fertilized, and you can get all the friendly non-native grasses that show up. Another threat to this habitat is scrub encroachment. And so this is kind of an interesting one, since it's native scrub, but it really is taking over a lot of the grassland habitat in California, and in many cases, it needs to get managed, and it is getting managed. On San Bruno Mountain, they pull out a lot of baccaris there. So this is just a cool picture that shows some of the shrub encroachment and the city in the background there. So it's kind of a neat picture. Another threat to the lupins here is this root crown fungus. And so this started affecting primarily Lupinus albifrons some time ago, and really kind of devastated some of the lupin populations. And that's had obvious negative consequences for the mission blue butterflies. And so we'll get into this a little bit more later, but part of the strategy then is to diversify the perennial lupin species that are out there that they'll use, more of the formosus, more of the variecolor, because they're less susceptible to this fungus, and that'll hopefully buffer against some of those kind of crashes. So here we are onto diversifying and amplifying lupins on San Bruno Mountain. So there's been a lot of work on this. A lot of people have been trying to build lupin populations on San Bruno Mountain for a long time. But it has been, most of it has been through trying to transplant plugs. And the problem with that is that you need nice deep soil in order to do it. And the places that the lupins tend to grow are actually really rocky soils. And the transplanting just doesn't work in those situations. And when you do plant in these sort of deep soils, then you get grass encroachment. And yeah, they can easily be outcompeted by grasses. So also, if you go out there with a lot of seed and you just kind of start spreading around, you tend to see really, really low germination, and so that hasn't really been a successful strategy either. Another difficulty has been that many of the local nurseries that have been trying to raise lupins for seed or for starts have had a really hard time growing them. They have fungal issues in the nursery, and it's been a challenge for a long time. So this is kind of most of the conservation community around the Mission Blue butterfly. The Park Service and San Francisco Rec and Park and San Mateo County Park, San Bruno Mountain Watch, everybody up here has played a role in this at some point. Basically everybody recognized this need for more seeds and better planting practices, really, to get numbers up. In come Disney. So, kind of an unusual funder, certainly our most unusual funder. We were able to get money through this Disney Butterfly Conservation Initiative, partnering with the University of Florida. And started working on these problems. So one of the first things we were able to do is we were able to really expand our nursery. We still have a very tiny nursery as compared to some of the nurseries I've heard around here, but we expanded it pretty greatly. You can see from kind of starting over here, and we have just all this bare ground, all the way through, and now we've got all these lovely beds and tables. We've got even more tables than that now. This is a nice boot wash station. This is this retaining wall that we built because when it really rains, it could flood through the nursery, and that's got a French drain, so we don't flood the nursery. We spent a lot of time building this up really nice for this project. And we've had a lot of success. We produced a lot of seed, a lot of really high quality seed, and one of the primary reasons for that, I think, is really the fact that our nursery is in Morgan Hill. So almost all the nurseries that were growing this were growing it really in the heart of the Bay Area, where there's heavy fog, and they were having a lot more fungal problems than we do, just because it was a lot moister there. We irrigate these so the plants get lots of water, but they don't always have so much water on their leaves. Another reason we were able to have success was these fairy bags. So this is Jimmy Quinnell over here. The nursery happens to be on his property, and this is his daughter, right? And don't worry, it looks like child labor, but she gets paid a fair wage in candy. And here she is, watering some of the lupins. But really, I wanted to point out these bags that we have here, these bags. And so what we realized when we started growing these that harvesting seed was going to be a problem because when lupin seeds mature, they shoot all over the place, right? And so one of the techniques that people have used to be able to harvest them is they harvest them a little bit green before they blow up. And we kind of felt like that wasn't really giving the seed enough time to ripen. We think that it has implications on how long the seed can be stored. So we got these They were advertised as fairy bags, so I'm going to call them that forever. But they're basically just these little fabric bags and with a drawstring. And once you can see that an inflorescence has gotten pollinated, or it looks like it probably has, it's far along, we'll tie it up. And then later, we'll go back and we'll snip all those out and we'll collect the seeds out of them. Unfortunately, they also add considerable time to the process. Having to go through and bag all those inflorescences and then take them, cut them back, and then go through them and sort out all the seeds is pretty time consuming. So it's definitely a boutique kind of method. But yeah, since 2019, we've produced over 150,000 seeds. And been able to provide seed to a whole bunch of different agencies, as well as our own experiments, which I'm about to show you. And we get viability of 99+% when we recently took some of our seed and sent it off for viability tests, and we were happy to see that. Oh, and see Jimmy's poster out there. That's important if you want to know more about the nursery and those operations. So, planting techniques. We knew we really wanted to be in these rocky soils, these bare soils. There's not a lot of competition. The lupins love to grow there anyway. Road cuts. Road cuts are a great place for lupins to grow. And it's amazing what a lupin radical can do when these things germinate. I mean, some of the best success we've had is just really shoving a seed like into a little crack in a rock. And that radical can find its way in there, and it's going to do just fine. So there, yeah, shoving seeds into rock crevices, and that's a nice example of a plant that's growing pretty much out of the rocks there. And then, even in the flatter places, sometimes it's maybe just pushing it into a really hard, rocky soil, but making sure to kind of cover it with mud, making sure not to plant until the soil's nice and wet. This day, actually, back there, you can see those clouds. This was a great planting day because we managed to mostly stay dry that day, but right after we were done, it just watered everything in, and it was lovely. And then I'll get into this a little bit more later, but one of the things that we found was that areas with lower insulation do better. I'm sorry?
What does lower inflammation?
Oh, I'm sorry. It means places like north-facing slopes and whatnot that get less sunlight throughout the year. Mm-hmm. So we set up this randomized block planting experiment. We planted in these grids that you can see. We put in permanent markers, painted nails that set where the grids would go, so we could go back with the grids and see what had germinated later, and what had survived through the years as well. We varied the insulation, the amount of light they receive. We varied the area of the mountain that we put them, the species, and the scarification methods. We found that lower insulation in the eastern part of the mountain were better habitat. And I'll talk a little bit more about species and scarification in a minute. So this is our 2022 monitoring data. So this is the most recent. We go back and we count everything and we see what's there. And it's so you've got year 1, year 2, year 3. And it's separated also by the treatment. In the first year, you see that we had this coffee soak treatment. It didn't work very well, so we stopped doing that for future years. After that, for the next couple of years, we were nicking seeds, which is pretty typical kind of scarification for lupin seeds. And you can see that the nicked seeds did a lot better than versus the unnicked seeds. pretty much every time. And so saying that, though, we do think, and we do still use as a practice, we plant a 50-50 mix of scarified and non-scarified. And we do that to kind of hedge our bets. Those scarified seeds are really likely to come up that first year, but with the kind of drought years we've had lately, maybe you don't want that seed to come up that year. It might be better sitting in the soil for another year. And so I think that, there's a better strategy is to plant the mix and hedge your bets. But that said, even in the 2019 plantings, it still shows up that so far, the treated seeds did better. But that could be just also because because it worked that year, and other years it could reverse that pattern. But we do see that every year so far. And we've established 434 lupins to date. And then this is the total number of plants. This is sort of the same graph, but it's number of leaves. And I just kind of wanted to put this up there just to show that some of these are pretty decent size. And so, you want to look at the highest bar on here or something. That's, so that's 40 leaves, but that's in a square meter. That's out of in the entire square meter, not average per plant. But if you look back, there was about an average of, say, like 2.5 plants, so about 10% of the plants were still there. And 40 leaves per 2.5 plants, you're talking about, I don't know, something like 15 leaves per plant. So that's a reasonable plant. That's like a healthy plant, not just something little that's going to die right away. So they really are seeming to get pretty well established. And that's the leaves. And then this is broken down by species, because I wanted to just kind of first talk about the total number of lupins that we've gotten out there, but really we need to really be diversifying here. One of the first things you notice is there's only 1 year for the variecolor. This is Lupinus variecolor, and boy, that one is our hardest one to grow. If anybody has ever grown any Lupinus variecolor out there and has some tips, I'd like to talk to you. It's hard to grow. It's hard to get big. It flowers later than others, and we end up getting Genista broom moths that really destroy them. So anyway, we've got this 1 year, and you can see that the average number of luva in a square meter is really pretty low. So we're really working on trying to get those numbers up, because it's an important species to really diversify out there, but that's where we're not doing so hot. But what's cool is that we have been able to plant LUFO all 3 years. And, we're getting pretty decent numbers. We're at least in a square meter getting a plant established. And, these perennial lupins are big plants. 1 per square meter would be really dense. So, we're happy with those results. And then the albifrons, that's the most common that's on the mountain also, has also done well. And we've only really had that for 2 years. That one just took a little longer to really get going in the nursery. But all in all, we've been able to get quite a few. And numbers are high on the luau. You can see why there's a lot of luau on the mountain. Luau does well on the mountain. Those are some of the highest numbers that we get of all. And coolest of all, the mission blue butterflies are using them. So this is Lupinus formosus with cute little mission blue egg on it. And finding that, that's what makes all the work worthwhile, right? And that's what we're trying to accomplish out here. So, some of the lessons that we learned from doing all this is this careful direct seeding into, really finding good habitat, planting seeds one at a time into this habitat. it works well. It's cost efficient in the way that we get good results, better results than most people have seen with plugs, and certainly better than broadcast seeding. But it also does take a lot of time. It takes a lot more time than throwing a bunch of seed out there, and we'll talk about that a little bit in a minute. Scar feed seed does better, more quickly, but planting in combination, adds to the seed bank and hedges your bets. I hope that, you look at some of those bars and they were, some of those unscarified bars and they were lower. I'm hoping that, some of those are still going to come up. Those are sitting there in the seed bank. They're not gone. And, maybe we'll have some real rain this year and we'll see some of them actually come back.
I think I'm going to say thank you very much.
Oh, okay.
If there's any more comments, you can talk to you after.
Okay.
We're already...
Thank you for attending this session and to all of our presenters. And if you have any questions, you might approach the presenters now. Thank you.